This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode number 222, recorded on March 1st, 2013. Hello, everyone. I'm Vincent Racaniello, and this is TWiV, the podcast all about viruses. Joining me today from North Central Florida, Rich Condit.
Howdy, Vincent. How are you?
I'm well, but you were not in Freeport, were you?
I was not in Freeport. Alan Dove was in Freeport.
Sorry.
Where was I? I was interviewing medical students. That's what I was doing.
All right, that's fine.
So I was actually working.
We were all making fun of you, having fun.
It's okay. I'm going to have fun in the next 2 weeks.
I understand. Also joining us today from Western Freeport, Alan Dove.
Good. It would be good to be there, but I'm here in Western Massachusetts now.
Did you enjoy Freeport?
Yeah, it was a good time.
And we didn't know it was you. I mistakenly said it was Rich texting, but
yeah, actually, when I sent that, I was I had just gotten to the airport to come home, but I figured, hey, TWiV is on. I'm going to go ahead and text.
Yeah, we mentioned it. Yep, we did dutifully.
Yeah,
also,
but incorrectly cited.
My apologies. Not the first mistake I have made, nor will it be the last, unless I happen to die in the next 10 seconds.
No, don't do that.
Also joining us today from Ann Arbor, Michigan, Kathy Spindler.
Hi, everybody.
Hey there. How's life? Is it good?
Yeah, it's pretty good. We had a big, heavy snow, but it's kind of mostly cleared away. So,
how many inches did you get?
They said 4 to 7, but it was the rain on top of it and the trees breaking and power outages. You know that all pretty well.
I'm familiar with the phenomenon.
Wonderful stuff. Well, here it looks like spring is back.
Good.
It's nice today. It's kind of cloudy, though, but temperature is up there in the 8 degrees centigrade range. It feels like you can walk around with your coat open.
Yeah, we're getting into mud season here.
And it is March 1st. We're zipping through the months. That's it for February.
Yeah.
Good thing none of you guys were born on February 29th, because you'd be kind of hanging in limbo, right?
Oh, I'd be 1/4 the age that I am. That'd be good.
Well, what? Never mind.
Did we celebrate this? DNA is 50 years old.
How can that be?
The structure of DNA
I thought it was about 4
solved.
billion years old.
No, 60. It's 60. The
structure of DNA was solved
53.
50 years ago.
No, it's 53.
53.
That's 60 years ago.
60. Okay, fine.
That's
50,
the year
60,
I was born.
You know.
That's the year I was born, so I always track my age by DNA.
Apparently that paper was published on February 28th or something like that. So
yesterday. Cool. I didn't know that. I missed that. All right. Thank you for noting that.
Are its telomeres still the same length?
Not mine.
Yeah.
Mine are getting shorter.
Shorter.
Or all of ours are getting shorter.
Yeah.
You can't avoid it unless you want to transform, and then that's not good. We have some follow-up. You know, last time we didn't have any. Now we have a surfeit. Is that the way you say the word?
I say it's surfeit.
Surfeit?
I don't know. Alan's
I thought it was surfeit too.
Okay.
Whatever. What do I know?
We'll surf the follow-up.
Yes.
We had a note from Twitter. I wanted to wait for Dickson for this one. He's supposed to show up.
Oh, you could read it every time until
wait.
he show up.
I'll wait. Dave writes, thank you again for the TWiV mention. The models do work well, and I am now in the process of assessing the success. P.S. Lawrence University is in Appleton, Wisconsin, just south of Green Bay, home of the Packers. You know, Green Bay is a tiny town, and they have this huge NFL team, and they make a lot of money because the NFL is a monopoly. I just thought you might like to know that.
Yeah.
You know, every team makes the same amount of money.
Really?
So my son comes home from school the other day, my senior, and he says, Dad, what's the only known monopoly in the world? I said, I don't know. U.S. government? He said, no, the NFL. So I guess he learned it in class. All right, next
There
one.
have got to be other monopolies.
I am sure. But his teacher probably taught him this, that
Well, probably the only operating monopoly in the U.S.
Maybe. By the way, Dave, we had a pick of the week. His little 3D models. By the way, Rich and Alan, did you like the title of last week's episode?
That was quite good.
That was Kathy.
What was that?
When you're there, done that?
Oh, yeah. That was great, Kathy.
I was very impressed. Good job.
Well done.
Kathy, we had a talk today, and a guy put up a slide of Bunya viruses, and at the bottom he had Bunya there, done that.
Ha!
No kidding.
He quoted TWiV. He said this was last week's TWiV.
Cool.
And my chairman turned and smiled at me, which is about all that I will ever get from him. I shouldn't say that, but I doubt he's
Acknowledgement
listening.
in the department for your TWiV. Okay.
That's right. Next one is from Richard. This is Richard Elliott, who was featured last week in some ways. Hi, Vincent, co-presenters. Enjoyed your last TWiV about newly emerging Bunya viruses. A couple of comments. Firstly, SFTSV. It's really a dreadful name. It goes against the tradition of naming Bunya viruses after their place of isolation, and severe fever and thrombocytopenia are hardly specific to this virus. Think about all the arena viruses causing hemorrhagic fever. I don't know whether this was picked up previously on TWiV, but there was a report in NEG, New England Journal of Medicine, about another phlebivirus called Heartland virus that caused 2 independent human infections associated with tick bites in Missouri. Turns out that this virus is related to SFTSV. So we did talk about this.
Yes.
Did we know it was related to SFTSV?
I'm not sure we knew it was related to SFTSV.
Yeah, okay. Makes sense, though. They're all Bunyas. Regarding Schmallenberg virus, our group did indeed establish a reverse genetic system. Oh, he said the word.
It's okay. You can handle it.
The old-fashioned way, by high-fidelity, long-range RT-PCR to make full-length clones, rather than Massimo Palmerini's synthetic approach that was published online in J General Virology at the end of 2012. We also showed that a recombinant virus lacking NS is a good interferon inducer. I would say that the old-fashioned way is the way I did it, which is to purify viral RNA and reverse transcribe it and then make it double-stranded with the Clenal fragment of DNA polymerase, and then attach GC tails and then clone it into PBR322 linearized with PST1.
That's the ancient-fashioned way.
Yeah, I'm an old guy.
Yeah, but something about PCR being called old-fashioned just does seem a little...
Kathy was right when she said I was moving to Glasgow, or rather returning to Glasgow. The lab moves later this month. Did you know he was moving back?
Well, I had some inclination from something I heard elsewhere. Yeah,
because you knew he had been there, right?
I thought that's where he was or was going. I don't know.
And then he was. Did you know he was at St. Andrews?
Not until you guys said that.
Okay.
So, yeah.
That's Richard Elliott, who's in St. Andrews and moving to Glasgow. And then the next email is from Massimo. I trust everything goes well with you. It was nice to hear you discussing our paper on Schmallenberg. I wanted to clarify a couple of points. One, Richard Elliott has developed independently a reverse genetic system for SBV. He has a paper that has been published online in December in JGV, and I guess will come out soon. Richard, as you know, is an excellent molecular virologist. He is so good that we recruited him back to Glasgow. So Kathy was right. Please pass my best regards to Kathy, too. Kathy, I'm passing his best regards to you.
Yep, thanks.
Finally, you might be interested to know that our PhD students and postdocs have awarded the 2013 Sir Michael Stoker Award to Francois Barré-Sinoussi. So you are in good company. Massimo. I got that award last year. I visited Glasgow and met Massimo. It was fun.
Cool.
So while I might have been right about Richard Elliott being in Glasgow or on his way there, I did make a mistake last week that I wanted to clear up. I referred to TWiV 188 as being the one that Richard Elliott had been on. In fact, he was on the one number 177 from Dublin, and that's when he talked about Schmallenberg. And 188 was the one from Glasgow that included Massimo talking about JSRV. And anybody that checks out the TWiV website and the archives would figure that out. But
can you read Suzanne's email?
Suzanne
please?
writes, I heard an interview with Dr. Oz from several years ago where he talked about bringing more aspects of spirituality into the hospital setting. I was honestly impressed. No, wait, hear me out. With many of the things he said. At the time, though, he seemed to be saying that it was a necessary human social side of interactions patients might need in addition to the medical side. I think that's a laudable goal. I don't watch his show and I don't keep track of what he's saying these days. I don't know if his views have changed or if I was hearing things through the filter of the interviewer's questions. But if his focus on spirituality now means replacing means-tested methods or even just adding to them when those additions might be dangerous, that's a sad thing indeed. Somebody needs to direct him to the quack cast. Or, just tested methods. Not sure why my brain added means in there. Politics and economics on my mind, possibly. So that was in reference to your pick last week. Right?
Yeah, that's the one that
Dr.
was
Oz.
criticizing Dr. Oz for being something beyond medical, yeah.
Back when he was a doctor, he apparently was pretty good, but now I think he's just totally gone off.
Well, yeah, and that was what this article was that I had cited. And now it's true that physicians do need a nice side, but diagnosis and medical treatments need to be founded in reality, right?
Yeah, yeah, and it shouldn't be about hawking the latest fad that's completely untested.
Right. Hey, Dickson, you're here.
I am.
Welcome.
Thank you. Sorry I'm a little late.
Ooh, if Dickson's here, can we go back and read this?
Yes, we have some follow-up for you, Dick.
Yeah,
Oh, wonderful.
we have an FU for you.
Really?
This came in from Tim via Twitter.
Come on, Tim, lay it on us.
In TWiV 221, Dickson was in his full form, funny and breathing loudly into the mic.
Oh, dear.
Not sure whether to find this endearing or irritating.
I think it's both.
Hey, can you not breathe this time?
I'll exhale only.
I don't.
It's more endearing if you play the podcast at 2x speed.
Right.
I'm sure.
Right.
All right.
Sorry.
Kathy, I screwed up. I meant to have you read your brother's email.
Oh.
Well, she could do that too.
Good.
That's
Why
okay.
don't you do that?
Sure. Bill writes, Kathy, Vincent, Dickson et al. While listening to the first discussion involving ticks and thinking back to all of the ticks I found in my apartment while living in deer-infested rural Central Virginia, 2010 to 2012, the statement was made that ticks were everywhere except Antarctica. My immediate thought was, really? I knew that there had been research on midges at Palmer Station, one of the places I've spent time at. They are insects, also arthropods. And then, surprisingly, the second discussion involved midges. So after getting home from running, when I usually listen, I had to investigate. And sure enough, there are ticks. They are parasitic upon seabirds, not mammals, in the warmer parts of the continent, basically the Antarctic Peninsula and surrounding islands. The significant species is Ixoxides uriae, known variously as the seabird tick or the Antarctic tick. I found several papers, and he gives a reference to a brief one, which describes their distribution along the Antarctic Peninsula. In Figure 1, Humble Island, Location 6 is about a half kilometer offshore of Palmer Station, and I've been there. Also of interest is this 2008 Antarctic Sun article quoting penguin researcher Ron Naveen. It's a terrible thing to see, Naveen said. The ticks get into adult penguins' feathers. The penguins start scratching. They stop paying attention to the eggs. We don't have an answer to this question yet about the timing of it all.
Wow.
Back to practical matters, this CDC site says while ticks as a whole are worldwide in distribution, most species are restricted to various regions. All major biogeographic regions, except Antarctica, have tick species of public health importance.
Uh-huh.
As for the midges, the ones which inhabit the Antarctic Peninsula are Belgica antarctica. They are flightless midges. At 2 to 6 millimeters,
Right.
they are the largest purely terrestrial animal on the Antarctic continent and the only insect, says Wikipedia.
Right.
They eat bacteria, detritus, and algae and have a 2-year life cycle. Antarctic Sun article is here, so he's given another link. With both of these parasites, warming temperatures and tourists are spreading them southward, and there are reports of invasive species as well. This was fun. Thanks again for attempting to stimulate this construction engineer's interest in smaller things. Bill.
But I wouldn't call the flightless midge a parasite.
You guys be careful what you say.
I know that if it feeds on bacteria and it has nothing to prey on because there's nothing permanent on that place.
Yeah, you're going to get another letter from Bill.
No, I won't. I won't. It's
Okay.
not a blood.
Well, if they're hassling these seabirds.
Well, no, the midges are eating bacteria and algae.
Right.
Oh, right.
Right.
So they're not parasites.
Exactly.
Yeah.
Exactly.
The ticks definitely are.
Oh, but of course. I don't know. There are no such things as free-living ticks. They're all parasitic or ectoparasitic. Right. So did we clear that air?
It's cleared. That's it for our FU. Let's move on to our two papers.
Excellent.
These were sent to Rich via Ed Niles, right?
That's correct.
Really?
And do you have a backstory there?
Yes, I have a backstory. So these papers come from the laboratory of James Chen at University of Texas Southwest Medical Center. And James Chen was a graduate student at the State University of New York at Buffalo in the biochemistry department during a time that both Ed and I were there. He was a graduate student with Cecile Picard, who was an absolutely brilliant biochemist who worked on one of the earlier workers on ubiquitin, and James's thesis was on ubiquitin. I can only say that I was acquainted with James. That was I left before he graduated, and I was gone on sabbatical for part of the time that he was there. Ed knew him well, and I believe was on his supervisory committee and helped them with a lot of molecular biology on some of their work, and is in fact on one of James's papers that came out of his PhD. And Ed has followed his career closely since then, and it has been an absolutely brilliant career. He is, as some of my colleagues describe him, on fire. These papers are, and has been for a while, these papers are proof of that. This will not be the last that we've heard of James. This is great stuff.
So on Monday, Dickson came in, and he had cannibalized his science issue on the weekend, and he deposited these two papers on my desk.
Right.
And
I did that.
And then, so Dickson, you got it right.
Hey.
These are two back-to-back papers in science. It's amazing.
Yeah.
It's just amazing. Now, I know Chen, I've emailed him a few times for plasmids. He always answers me right back. He sends them. He's really nice. I know him because he discovered IPS-1, which is an intermediate mitochondrial-bound protein involved in sensing of cytoplasmic viral RNA.
Wow.
In the innate sensing pathway.
That's same as MAVS?
Yeah,
or?
MAVS, IPS-1, Visa. 3 different groups identified it at once. And they all gave him different names.
Of course.
And I had asked him for a clone. He sent it right away. Seems like a really good guy.
Yep.
And they're probably all still fighting about what name is appropriate.
Yeah. So of the 3, IPS-1, MAVS, and Visa, which do you like?
I like Visa,
I'm
actually.
used to MAVS.
I use IPS-1. We actually published a paper on it, so I just use IPS-1.
But Visa is everywhere that you want to be.
It is.
This is all true.
If we did a podcast on that, we could have made that a title, Alan. Anyway, the overarching theme of these papers is looking for the sensor of DNA.
Right.
Because we know the sensors for RNA, they're toll-like receptors, they're cytoplasmic sensors. We know the pathways to induction of interferon. And maybe I should back up. Why do you need to sense? Well, when viruses, bacteria, pathogens infect, they're initially sensed by the innate immune system. So the sensors, cell proteins, sense them. So viral RNAs can be sensed by TLRs, toll-like receptors, and cytoplasmic sensors like RIG-I and MDA5. There are some sensors for other products like lipopolysaccharide glycoproteins, but the DNA sensors have remained moderately elusive.
And we're talking here about DNA that has to be someplace or doing something that it shouldn't be doing.
Right.
Because obviously there's DNA in all the cells in the nucleus, but in this case, if you have DNA in the cytoplasm, somehow it trips an alarm.
And in the mitochondria.
Right, and even DNA leaking out of the nucleus into the cytoplasm is a signal that things are not all right with the cell.
Right, and the cell responds to that.
Response. Yes.
Right. So the question is, how does the cell know that there's DNA in the cytoplasm?
All right, so this is a bone of contention in my view. All right. Now I know, Rich, that things are sloppy in cells. Okay? You've mentioned that many times, and I believe you. However,
Good. He's going to get crucified for this time, not me.
Yeah, this is the big however.
That's
all
right.
right, look, let's take an RNA virus. It infects, and its RNA ends up in the cytoplasm, which is where it wants to be. Let's say polio, because it needs to be translated. It gets sensed by RIG-I, which is right there in the cytoplasm. Okay? Many RNA viruses do that. On the other hand, with the exception of a few very large DNA-containing viruses, such as the one you work on, all the others go into the nucleus.
Right.
And so why are they being sensed in the cytoplasm? This makes
no
sense to me.
It's an excellent... Well, they have to go through the cytoplasm to get to the nucleus.
All right, so what you're saying is that they're leaking on the way in, and that's being sensed? I just don't buy that.
No,
well, I think that this is something that's going to have to be researched. Either that or somebody out there knows something and they'll tell us. But I think this is an excellent question.
As a naive person of this group, didn't you guys instill in me the concept that not all the virus particles that are made are competent? And what if you had an incompetent virus particle with DNA?
I don't buy that. I don't buy that the abnormal is going to be responsible for a major pathway like this.
Well, it's a way of detecting it, though, because
it
never
completes. Most of the DNA is in the nucleus, okay? So, and in fact, whenever you ask these people who work on DNA sensing, what's the story? Most of these DNAs work in the nucleus. They'd give you the leakage hypothesis. This is like the Cut-Hunter hypothesis. It's the leakage hypothesis that the DNA leaks, but I just don't buy it.
Well, thinking
not even late in infection when the nucleus starts to break down.
Yeah, but then it's too late, right?
Yeah. Well, except that the cells that are doing this are not going to are not going to survive anyway. All of this has to do with warning their buddies.
Yeah, this isn't a mechanism to save the cell.
I know, but the contrast between the RNA and the DNA is pretty big because with the RNA sensors, it's right there where the viruses are, right?
I don't disagree that this is a significant question that needs some attention.
Kathy, you were going to say something.
Wait. One other thing. It just occurred to me.
But you can't.
It just occurred to me that what happens during mitosis when the nuclear membrane goes away? I wonder... you basically have DNA in the cytoplasm.
Right. So then you don't want to make an immune response, although in some people you have autoimmune diseases as a consequence.
You do.
In most people, you don't make an innate response against that DNA. So
there's going to have to be some damping of this system.
Right.
Yeah.
Controls.
Okay, I'm done.
And what I was going to say was that for adenovirus, where the particle starts to come apart in the acidified endosome, it's not entirely clear to me that there couldn't be some DNA leakage right then and there. So you have I'm looking at a picture which I think I pulled up on Google Images, which I'm thinking is probably from Principles of Virology. And it looks like, that the capsid has some holes in it at that point. And that would make sense based on what we know about protein 6 and endosomal lysis. And so, to my way of thinking, at least for adenovirus, that's maybe where it happens, where and how it happens.
So that could be, but I'm still surprised that you're sensing something leaking as opposed to a major pathway. Then you take herpes viruses, which dock onto the pore, and then the DNA goes through, or the parvos, which fit through the pore intact and uncode in the nucleus. So I'm happier when there's a universal mechanism and not based on leakage, but
is this paper attempting to explain for all DNA containing viruses?
It says it's a sensor of cytosolic DNA.
But it doesn't say which one.
It tests two, herpes and vaccinia, and it does seem to respond to those viruses.
Right. Yeah, and what they're actually looking at is they're trying to figure out the mechanism for a documented phenomenon, which is if you have DNA loose in the cytoplasm for whatever reason,
right,
that triggers an interferon response.
I just think there's got to be a nuclear sensor. Okay,
maybe.
Because I think that the most of the DNA is in there, although you could argue that then how do you distinguish from chromosomal DNA? But there could be ways. It's all nucleosomed and viral DNA might not be. But anyway, that's my issue. These are wonderful papers. It's beautiful. That's my issue since you brought up this leakage issue.
If there is a nuclear sensor, my money would be on Chen finding it.
Yeah. Well, unless he's convinced that this is it and there's nothing more, Some people fall prey to that sort of,
I don't know. Just from looking at these two papers, he doesn't seem like somebody who just lets something drop.
No, he's not going to fall prey to much.
All right, so so many people have worked on this, and there have been a couple of sensors described. One of them is called Sting. All right, what's the name? What's the acronym for? I'm not finding it here.
It's in the
It
paper.
is the I had it highlighted here somewhere. Stimulator of interferon genes. Very creative.
Sting. Okay, Dickson, Sting. Sting.
Yes, sir.
And
Not the singer.
No,
but
don't you ever lose your faith in him.
I shan't.
but how Sting was activated by DNA in the cytoplasm wasn't known.
Right.
Okay, and
So it's kind of a middle of the pathway thing.
right. So, Rich, do you want to describe the assay they developed? I think you're really hot on this.
Okay. First of all, this is coming. There are two papers here, as we've described.
I'm
and
starting
I think
with
the
the
first
page.
one. The first one. We ought to do the one. The
826,
first one
right?
is the cyclic GMP-AMP paper.
Yeah, that's
right?
the first one. They put
They
them
both
out of
start
order.
that.
They put them out of order. That's right. And
Do you mean the is an endogenous second messenger
Yeah.
paper, or the
yes,
is a cytosolic
the is an
DNA?
endogenous second messenger paper. And the authors are, how would you pronounce this? Jiazi Wu,
I'm gonna take a guess at Jiaji.
Jiaji Wu,
Yeah.
Li Jun Sun,
Right.
Zhang Chen,
Right.
Feng He?
what?
Feng He, maybe?
Fanga Du, Heping Shi, Chuo Chen, and James Chen.
And we apologize for butchering your names.
Yeah, right.
At least you're getting a good bump.
So I think that the assay here is key to all of these two papers. So the question is, we know that in response to DNA, Sting is activated, and that triggers a bunch of downstream events that wind up inducing an interferon response. And it activates NF-kappa B and it activates IRF3. And importantly, the activation of IRF3 involves its dimerization. And one can measure the dimerization of IRF3 by running native gels so that the dimers are not dissociated and the dimer runs slower than the monomer. And you can detect this either by Western blot or by inputting radioactive IRF3 and doing an autoradiogram. And so they want to know what the steps in the pathway upstream between DNA and Sting are. And what they do to do this is basically uncouple the stimulation from the response with a I would call it a sort of a semi-in vitro assay. They start off with cells where they have artificially inactivated Sting, so they will get only activation. And the way they artificially inactivate Sting is with an shRNA construct. So it's an RNA interference thing, and they've knocked down Sting. And they then activate those cells either by transfecting DNA or infecting or whatever, but they introduce DNA somehow and activate those cells. So presumably those cells now have whatever is upstream of Sting in them. They assay for that by making a cell extract. So they break the cells, they spin out the big stuff. They have what we call an S100, which is the supernatant from 100,000 G centrifugation. So it's a soluble extract. And then they add that to cells from another cell line where those cells have been permeabilized with a porphyrin. Porphyrin is either a bacterial or a yeast protein that is a toxin that's made by yeast or bacteria, and that will bind and invade membranes and form pores in cells. That's how it kills them. But it also can be used in this context to let big things in. When you permeabilize cells that way, some cytoplasmic stuff comes out, but they knew that sting is attached to the endoplasmic reticulum, so it's not going to come out. So they take this extract from the stimulated cells and they use that to treat the permeabilized cells, and then they look in the permeabilized cells to see if IRF3 is dimerized. And sure enough, they can show that if they stimulate cells and make an extract and then assay that extract by looking for IRF3 dimerization on a target cell, they can see that happen. So this is assays are everything, and this that's their assay, and it's used throughout both of these papers. So now they have an assay for whatever it is that's upstream of sting, and they can go ahead and try and purify what that is.
Cool. Yeah.
Right.
And one of the early things they do is to heat the extracts to 95 degrees. What do you think of that, Dickson?
That'll precipitate the proteins.
It'll denature if it's a protein,
That's
right?
That's the first thing anybody does when they're
Yeah.
doing this. They say,
Yeah.
we're going to prove that it's a protein
Right.
Or not.
by denaturing it at 95 degrees.
Right.
So they find that 95 degrees doesn't affect the activation of IRF3. They treat it with nucleases, so they say it's neither DNA nor RNA. Proteinase K, which digests proteins, doesn't get rid of the activation. So they
Right.
say it's probably not a protein, DNA, or RNA.
And I would say that if it was me doing that, I would say that was both a good news, bad news situation.
Exactly.
Because
The good news is that you can boil this crap and
Still get it
most
to
of
work.
everything will precipitate and you can just spin it out. So that's probably 100 or 1,000-fold purification right there. The bad news is that how are you going to figure out what this teeny heat-resistant molecule is?
Well, that didn't stop James
No.
Chen, right? It would have stopped me. I would have said, I don't know what to do.
Yeah, okay. We're done.
Gas chromatograph.
We have a heat-resistant something.
So then they have to identify it, and really this is quite an involved thing, but they basically use modern technology.
Mass spectrometry.
Mass spectrometry to identify what is active in inducing IRF3 dimerization. It turns out to be cyclic GMP, which is a molecule that you love, Rich.
I had never heard of it until I saw this, but I love it now.
Yeah, they have a wonderful discussion, which we don't have time to go through, about how they honed in on this, and they had a lot of clues. That's it. Cyclic GAMP. And you put a nice picture of it in the show notes here.
Yeah, so that is it is an AMP and a GMP,
Right.
and each is linked to the other by a 3' 5' phosphodiester bond.
Right. Cyclic, right. So very much like cyclic AMP, except this time we have two bases
Right.
involved, bases and sugars. And you'd never heard of it. I'd never. Although
I'd never heard of it.
In bacteria there is a di-GMP signaler, I think, and they mentioned that here. Let me
Right,
see
and a di-AMP, and,
Right.
and knowing those things, and they knew that, and they knew that those things stuck to sting. So I think that was a big clue.
Right.
And it was also interesting that the mass that they got out of their mass spectrogram was, I love this, the mass that they got was an average of the masses of dicyclic AMP and dicyclic GMP.
Right.
Sounds like
Right.
Erwin Chargaff.
Right. So, yeah, in fact, they say that this molecule is the first cyclic dinucleotide in metazoans.
Right.
Right.
So, metazoans being things with a stomach.
Is that right?
That's my definition.
Things with a
That's
stomach?
right.
No, it's just
Yeah.
multiple cells.
Things with a
Yeah, I
stomach.
thought it was multiple cells.
Playing a Midwestern joke.
I believe that's true.
Also known as animals.
Yeah.
So I'm a metazoan, right?
You're a
Yes.
multicellular
You're a metazoan.
organism.
We're all
Yeah.
metazoans.
Protozoans are
So a plant
not?
would be multicellular, but not be metazoan. Right?
I don't know about that one.
Yeah, a plant is multicellular, but not a metazoan.
You don't use the word zoan.
Right.
You use another word. Phyto. Well, they use zooplankton and phytoplankton to distinguish the kinds of plankton. So maybe metazoan and phytozoan. Maybe it's a phytozoan.
Okay.
So Wiki says animals are a... Oh, no, that's animal.
Wiki, yeah. The world's greatest authority. I think if we asked Lynn Margulis, she might be able to
Dad,
tell
she
us.
can't tell you.
Yeah, I know, but that's the way it goes.
Well, look, I don't share your lack of enthusiasm for Wikipedia.
So
It is a crowdsourced thing, after all.
Can I ask a stupid question here? And that is,
sure.
I was under the impression, since you were talking about DNA viruses mostly locating to the nucleus during their replication cycle, how does interferon work in that sense?
Many ways.
In the nucleus?
Well, there are many ways because it induces over 1,000 genes, each of which has a different antiviral activity,
Right.
most of which we don't know. So
I see.
some may
I see.
target nuclear function, cytoplasmic.
So it doesn't act directly on viral replication. It acts
Well,
by inducing things that act directly
Yes,
on viral.
exactly. Interferon themselves have no direct antiviral activities. It's the proteins induced by them.
Poorly named.
Metazoan, any of the animals belonging to the subkingdom Metazoa having a body made up of differentiated cells arranged in tissues and organs.
There you go.
Okay.
All multicellular animals besides sponges are metazoans.
Right.
And it's specifically animals.
Yeah.
Anyway, I think it's pretty cool to discover a brand new compound in this day and age that
Here,
no
here.
one's ever seen before in
that's true.
metazoans.
You're
Isn't that
absolutely-
cool?
Yeah.
Yeah.
I
Yeah.
mean, you know, many people now training to be scientists think everything's been discovered. But this shows, in fact...
Not even close.
Not even close. In fact, the gene in the next paper wasn't even suspected to do what it's going to be shown to do. So then they do a lot of experiments on this cyclic GAMP. CGAMP, like putting it into cells itself, they synthesize it and put it in cells and show then that they make interferon. I think they show that ATP and GTP are both needed, right? So you can make this. And that's a clue. Something must be making the CGAMP at Dickson, right?
Yes. I'm still following this.
DNA virus infection, herpes simplex virus infection leads to the production of CGAMP in cells, but not VSV infected, which you would not expect, right? It's a good control.
VSV, you get IRF3 dimerization, but it's not mediated by CGAMP.
And that's, in fact, the sensors of RNA that we talked about earlier, they work through MAVs, VISA, IPS1, and they end up phosphorylating IRF3, and you get dimerization, just as STING leads to IRF3 dimerization.
Right. So this is a DNA-specific signal.
Right. They even use vaccinia virus.
Hooray!
Do you think he got it from you or Ed Niles? Or
No, I don't think so. Well, he might have gotten it from Ed.
There are a lot of ways to pick up vaccinia.
I don't think I sent him any vaccinia.
Anything else we should discuss for this papers before we move on? The last experiment examining whether STING binds to CGMP directly. Right?
Right.
Yeah.
That's important. They make P32 CGAMP, and they cross-link it to E. coli that are expressing STING. There's just so much in this paper.
Yeah,
Wow.
it's huge. Actually, another really impressive thing is they have a chemical synthesis of CGAMP. They make it themselves.
Right.
Oh, yeah. It's in the supplementary figures, right?
Yeah. I think that's why there's those two added guys with the superscript 3 in the author list.
Yes.
They're the biochemists.
That's right.
And also, then when they make the P32 labeled CGAMP,
Yeah.
they use the Vibrio cholera enzyme to do that. So they use the bacterial enzyme, which I thought was kind of cool.
Anyway, they show that it binds to STING, and so that's how they think it activates it, by binding to it. So, Dickson, do you know any other known functions for cyclic nucleotides?
Well, they're involved in intracellular signaling,
Do you know an example of one?
cyclic AMP.
Good. I knew you would know that.
We didn't rehearse this either. This is
That's
a de
old
novo
school.
answer.
So this is like another second messenger, right? Basically,
Yeah.
It's- Somehow it's being induced
That's right.
by DNA leaked into the cytoplasm and
Right.
it's binding STING, and that's inducing interferon.
Very interesting.
I have to say a very sad thing. I have a colleague here who worked for many years to identify a DNA sensor, and then a couple of years ago, Sting was published. So he got scooped, and now with this, he got scooped
You
again.
mean he got stung?
Yeah, poor guy.
Jay.
Yeah.
Yeah, ouch, twice.
One thing I had as a question, and I don't know, it seemed like it was this paper where they tested the different kinds of DNA more thoroughly than the next paper. Do you guys have any sense for whether single-stranded DNA can
Good
activate?
question. Single versus double-stranded, right?
I didn't see any single-stranded DNA substrates in any of their stuff, but
Right, I don't know. I do know that they use this special
Yeah.
oligonucleotide called ISD, interferon stimulating DNA. That I looked it up, and my understanding is that it is specifically devoid of CpG dinucleotides because those trigger a different pathway.
Yes.
That's right.
in the supplement for this paper, they talk about it being prepared from equimolar amounts of the sense and antisense DNA, and then they list it. And so that's what clued me in that it was double-stranded, because for a while I thought, well, maybe that's just a single-stranded substrate, but it's not.
Yeah.
So I do not know.
Yeah, all the substrates they list in the beginning of the paper, they have this ISD, they have a 50 base pair GC-rich double-stranded DNA, they have poly-DIDC, and herring testis DNA, all double-stranded.
Well, this all goes back to our initial discussion, because there has to be some way to discriminate between your own DNA and DNA that shouldn't be there.
Right.
And whether that's just because of the compartmentalization or whether there are other issues involved, I don't understand, at least. Maybe somebody does.
All right, I have a question. There's an accompanying perspectives article by Luke O'Neill,
Right.
and it's got a nice title. It's called Sensing the Dark Side of DNA. And that refers to foreign and mislocated DNA in the cytoplasm. And they have a picture where they show DNA, double-stranded DNA, coming through the nuclear pore complex and just going out into the cytoplasm. Now, it's my understanding that at least RNA has to be transported out. We talked about that a couple of weeks ago by the exportant system. So what's with DNA? Can it just leak out, or does it have to be transported? Does anyone know?
Well, it's certainly not supposed to leak out,
No.
Yeah.
except during mitosis when the nuclear membrane breaks down.
I
think
that's
my
the big
my reading of that figure was that was being depicted as an accident.
Yeah, that was my reading as well.
because there's no discussion of.
But if it did, for whatever reasons, there would be no consequences if it was host DNA, right? Because the immune system is directed against the virus, and
Right, but if it triggers this system and stimulates the interferon response,
then you can get inflammation.
you could get inflammation and possibly an autoimmune disease. And in fact, the perspective article goes into that as a possibility that this could be part of the mechanism behind things like lupus.
And they've also got part of that diagram. My interpretation was they got DNA coming out of mitochondria as well.
Right.
So, my interpretation of this is if there is DNA coming out of the nucleus or out of mitochondria, there is trouble. Okay,
Right.
and it's time to close down the shop.
Okay. All right, the second paper is in the same issue by the same PI, and it's called Cyclic GMP-AMP synthase is a cytosolic DNA sensor that activates the type 1 interferon pathway. The authors are Sun, Wu, Du, Chen, and Chen. And I just avoid the first names.
Well, they were all, we listed all of them previously because...
We tried to.
Yes, we tried to, because the previous paper that we talked about is all the same people plus a couple of biochemists.
Right. So here they try to purify that which makes CGAMP. Right. And they use the assay that Rich described earlier, putting extracts onto these perfringinolysine O permeabilized cells and then look for IRF3 dimerization. All right. So they take the extracts and they begin to purify them. And here is where I am knocked over. They use 3 different methods of purification.
Yes.
And
I couldn't get one to work.
so I'm thinking, any other lab would use 1. They wouldn't see anything in the end and give up. And these guys used 3. And in the end, they couldn't identify any single protein that co-purified with the activity, which is the C-gas activity, which is to make cells dimerize IRF3, right? So
and in each case, they're starting off with 200, 150 millimeter dishes.
oh my gosh.
Wow.
That's and that's for their final prep in each case. You know that they spent time messing around with this. 200, we call those frisbees, the big guys. 200 frisbees is the equivalent of 10 liters of HeLa cells.
Amazing.
Heavens.
So then they took these 3 purification methods, and they took the last step of purification and did mass spectrometry to see what was in them.
Right, so they've got the purest prep from each of these. They still don't know what their protein is, but they ran mass spec on the final fraction.
And they say, although many proteins co-purified with the C-gas activity in 1 or 2 routes, only 3 proteins co-purified in all 3 routes. And this is by mass spec picking them up. You can't see them on a gel.
Right.
This is phenomenal.
Yeah.
Can't imagine what this cost. And all 3 proteins were uncharacterized. So this is the cool part. They're uncharacterized proteins. None of them, nobody knows what they do. So they said bioinformatic analysis drew our attention to E330016A19.
That's easy for you to say.
Which had homology to a protein called oligoadenylate synthase.
Oh my goodness.
Which is an interferon-induced protein involved in the RNA cell pathway.
Wow.
And they basically end up showing that this is their enzyme that makes C-GAMP.
So I want to say a couple of things here. First of all, about the purification.
Yep.
If you're in the old days, if you're lucky, you could put together a nice simple assay. So you took your column fractions,
Right.
you just added them to a tube, and in about 2 seconds, the tube turned purple or something like that. If it had your assay, this is the assay we described, where they take these fractions
It's a live assay.
and stimulate permeabilized cells and look for dimerization of IRF3. So it's an extraordinarily laborious assay.
Right. Run it out on a gel, blot it.
Yeah, the whole thing. I mean, so that's a lot of work. Second, this idea of the 3 different purifications. As you're developing a purification, it's not uncommon to go through and say, well, that didn't work. What if we switch these columns around? Or let's add this column. So you're likely to develop 3 or 4 different purifications or possibilities as you're going through a purification. And then you wind up and you look at the gel and there's a smear of proteins and you say, well, that's not good enough. The classic way is to add a few more columns, right, and try and clean it up more. The real innovation here is to take 3 different methods that didn't really work and individually all that well and do the mass spec and find out what they have in common. And they say in their discussion that's a novelty. That's the beginning of their discussion. And I wholeheartely agree. My guess is that this is going to become not an uncommon way to purify proteins. This is, adding the mass spec in as the modern addition to purification. It's brilliant.
And my guess is that this grew out of hitting a wall.
Sure.
That they did a purification, they got to the end of it, they couldn't get a co-purification, they couldn't figure out what the molecule was at the end, but they've got this final fraction that's got the activity. Okay, let's do a different purification, start over. And then they did that, and after three times through it, they still weren't getting the answer they wanted, so they looked at the whole process and said, well, let's analyze our final fraction from each of these and see if anything pops out. It strikes me as kind of I don't know if this was true, but it strikes me as maybe it was a move of desperation. Let's just see what's in there, and they ended up with this.
Well, I think he knew there was an activity that made this, and he was he was
just
determined to find it. Right?
There was clearly an activity that made it, and they just did not give up when any sane person would have given up.
The other thing that I can imagine is the excitement to find in this homology to OAS1.
Yeah.
Because the parallels here are striking. OAS1 is a nucleic acid sensing protein that's involved in stimulating the innate immune response. It binds nucleic acid, double-stranded RNA instead of DNA, but still, and when it's activated, makes a weird oligonucleotide. Okay, in the case of OAS, it makes 2' 5' oligoadenylate, which is probably even weirder than CGAMP. I don't think any of that's a coincidence. These things have a common ancestor somewhere back there. The evolution of these things is fascinating.
Yeah, so they saw that and they said, this is it.
Yeah, this is it.
And they did a whole bunch of experiments to show, they overexpressed it, they knocked it down, they showed expression in different cell lines correlated with C-gas synthesizing activity. You need this enzyme to make interferon to induce it in response to virus infection and DNA introduction into the cytoplasm.
They mutated it.
They mutated it. They located what part of the protein was important. They showed that you can make recombinant protein. It will make CGAMP from ATP and GTP in vitro, but only if you add DNA to it.
Wow.
Which is pretty cool. This is a DNA sensor.
Yeah, so this is the thing.
Senses DNA, and then it makes C-gas from ATP and GTP, which is really unusual, right? We don't know of any sensor that does that so far.
Right.
It's very cool. And it also binds to DNA, so that's cool. That's how it senses it.
Right.
But again, Kathy, no single-stranded DNA here, right?
I don't think so.
It's interesting. And cytosolic localization. Yes. So this enzyme, C-gas, is predominantly localized in the cytoplasm. Barely detectable in the nuclear extract. So this is not your nuclear sensor, if there is one. However, listen, the protein could also be observed in the nuclear or perinuclear region. Okay, so maybe it could hang out right at the pores and somehow sense DNA inside. Who knows? I don't know. Something going on there. Anyway. Okay. So these are, then you can look at all the sequences of everybody that have been determined, and you can find this C-gas that's in all kinds of metazoans. Actually, also in they say the large family of C-gas that is conserved from fish to human, formally demonstrating that vertebrate animals contain evolutionarily conserved enzymes that synthesize cyclic dinucleotides, which were previously found only in bacteria, archaea, and protozoa. So this is really a tour de force.
It is.
Yes.
It's amazing.
It's beautiful work, and it opens the road up for a lot more things to do, including find out what the nuclear sensor is, I think. But really, there are DNA viruses that may bypass this, so maybe there's something else. Anyway, Dickson, nice papers, don't you think?
I love them, yes.
Any questions?
Tons of them.
Give me one.
Well, I want Kathy's question answered, for instance.
Single-stranded DNA?
Betcha.
Okay. But you can't take someone else's question.
Yes, I can. No, I'm a follower. I'm not a leader in this field. Trust me.
How about, what are the viruses doing about this?
I'm sure they're doing something, right?
There have got to be viral proteins that target C-gas.
That is an excellent question. I like that one, Alan.
Sure.
Every DNA virologist who's interested in innate immunity has to be now picking up this protein and looking at it, right?
Well, in fact, when they do their herpes infections, they use a herpes mutant that is crippled with respect to immune defenses. I forget exactly what it is or what it does, but they start with a herpes mutant that already is a little crippled in this regard.
Yeah, that in fact was one of my questions. It's a deletion in 34.5 gene, and rats. I can't find exactly where it describes it. Figure 3D.
Top. That's the other paper, I think.
Yeah, it's in the other paper. A viral protein known to antagonize interferon production in the infected cells. So I wonder if they had to use that because a wild-type herpes would have messed up their assays too much.
That's what I
Could
guess.
be.
Yeah.
And I was actually even surprised that they got away with just using wild-type vaccinia to do this, because I would think vaccinia would be loaded with stuff that would counteract this, but maybe not enough. I'm sure we'll hear more.
Dickson, here's one for you. So they think that maybe this could sense malaria in the cytoplasm. Does that make sense, Dickson?
Well, malaria, you mean in the hepatocytes when it enters first?
Is their malaria DNA going to be in the cytoplasm where it could be sensed by this?
Well, it's surrounded by a cell membrane, though, you see. So it's a protozoan living in a metazoan.
Maybe it leaks, Dickson.
Maybe. I know, I'm game. But malarial parasites, once they're in the peripheral blood, infect red cells, and whether red cells would have this same system or not, it's problematic at that point.
They say retroviruses now, those guys actually start reverse transcription in the cytoplasm in kind of a subviral particle, so maybe some DNA is accessible to.
But Toxo could do this. Toxoplasma might very well fit into this pattern.
Hey, Kathy, maybe in AD, this thing can actually get into the particle and sense the DNA in the particle, right?
Could be.
That could be.
If it can get into, I can't think of the word, permeabilized cells.
Yeah, right.
Yeah.
All right. So there you go. Yeah. Very cool papers. Thank you, Ed Niles. And thank you, Dickson Despommier.
It was sheer curiosity as to what you guys would do with it. I'm amazed.
And thank you, James Chen et al.
Yes. Absolutely.
I just can feel his enthusiasm.
Yeah.
Not giving up and telling his people, come on, we have to do this. I mean, you can't do this in a lab with one person, obviously.
No way.
Right.
But
no way.
It's just great stuff, and it just shows why. While we were talking, I got a little notification on my phone. It says here, "Oh, come on, phone! U.S. government to begin steep spending cuts after Obama and congressional leaders failed to reach deal on sequester."
Oh yeah.
Here we go. And science is going to be. It's good stuff is going to be stunted as a result. It's really sad. Okay, let's do some email. Alan, could you read the one from Tim?
Sure. Tim writes, I've listened to your podcasts on Science 360 for over a year while traveling and continue to do so here on internet radio. And now, since I can easily listen to your podcasts web-wise and re-listen online to reinforce what I hear. This area of knowledge isn't my forte, but I do love learning, and yourself and your guests give me lots of material and motivation to learn, even at my old age now. My background is IT, Unix, TCP/IP development in the 70s and 80s. I've always had an interest and love for what biologically makes us live and the practical issues faced by doing so. Your podcast conversations motivate me to learn further, and in the capacity that I can, I do, and I thank you a lot for that. This is just a note of thanks to yourself, your regular and other guests, and entire team that make it happen. Tim, who's a listener in Denver, Colorado.
Nice.
Well, thank you, Tim.
Excellent. Indeed. Thank you very much.
Rich, can you take Daniel's?
Sure. Daniel writes, I guess for every, Letters to a Young Scientist by Edward O. Wilson. Wilson channels Rilke in this epistolary series meant to inspire future generations of creative, curious scientists. He articulates why good science is so necessary to civilization and how aspiring scientists can best take advantage of their own research interests. Wilson's own personal anecdotes from a long, distinguished career add richness and depth. So for every of those, there will be a Bad Pharma, How Drug Companies Mislead Doctors and Harm Patients by Ben Goldacre. A journalist and physician, Goldacre focuses his ire on the pharmaceutical industry's deceptive practices from the research laboratory to the doctor's office. Writing for laypersons, he shows how much research is faked, statistics are twisted, and doctors are compromised by a horribly disfigured web of money and political influence. And Daniel continues, your wife must be so happy. I'm sure it will be objective and fair. So, yeah, I saw this and I thought, well, almost every human activity, if not every human activity, is corruptible.
And it's not a horribly disfigured web of money and political influence. It's a finely engineered web of money and political influence. But yeah, I mean, actually, Ben Goldacre does have a point. I haven't read the book. I don't know if he carries it too far or if the title is a bit sensationalized. But
there's more about this down further on, I think, about Gold.
Okay, yeah. I've followed some of his work, and he actually, he has a lot of good and useful stuff to say, calling out quackery and criticizing things that should be criticized. So I don't know if he's still on target or if he's going over the edge.
Well, for all the bad things that pharma does, they do some good things.
Mm-hmm. Sure.
Yeah, absolutely. Mm-hmm. Absolutely.
And that is the mechanism we have for making new drugs. So,
right.
If you're going to criticize it, as I think people should, and think about how it's done, I think there's also an obligation to come up with a better method.
All right. Kathy, can you take Peter's?
Mm-hmm. Peter writes, Dear TWiV team, from a cloudy, cold, and damp England, 4 degrees Celsius, humidity 87%. Not directly related to virology, but I thought that this may be of interest to you and your listeners. The All Trials campaign, and he gives a link to that. Campaigning for the publication of the results, that is, full clinical study reports from all clinical trials, past, present, and future, on all treatments currently being used. GSK, so that's GlaxoSmithKline, have announced that they are signing up to the alltrials.net campaign.
Wow.
Dr. Ben Goldacre, who helped develop the All Trials campaign, is also campaigning for true evidence-based social policy from government, and he gives a link for that. And then, so that relates to what we were just talking about. And then he has some questions about viruses. 1, are there any retroviruses of prokaryotes? 2, are there any retroviruses of protozoa? 3, are there any phages that infect bacteria or chloroplasts? I assume they are protected by being inside eukaryotic cells.
I think he probably means mitochondria.
Mitochondria.
Oh, right. Yes, absolutely. Yes. And then 4, as mitochondria and chloroplasts evolved from engulfed prokaryotes that at one time existed as independent organisms, is there any evidence for ERVs, endogenous retroviruses, in the mitochondrial or chloroplast genomes? Regards. So, first question, are there any retroviruses of prokaryotes?
I don't know of any.
I don't know of any.
I mean, there are phages that integrate into their genomes, right? But those are DNA viruses.
But they don't replicate through an RNA. I don't know of a prokaryotic reverse transcriptase.
No. Right.
Although there is reverse transcriptase activity in some bacteria.
Okay.
But I'm not aware of any virus carrying it. They may be from those transposable elements that move around.
Right? Good point.
But I don't know of viruses though that carry.
Yeah, I don't know of any retrotransposons in bacteria, though I haven't thought about this for a long time. Most of the transposons are entirely DNA based and replicate through. Sorry. Go ahead.
No, I was just going to go on to the next question.
Yeah, I'm done.
Oh, okay. Are there any retroviruses of protozoa?
Dickson?
Yeah.
I'm the last person that would know the answer to that, but we can certainly query the audience on TWiP.
Right.
I don't think there are any.
There are very, very few viruses known to infect protozoans, and most of them, as far as I remember, are double-stranded RNA viruses.
Yeah, I don't know of any in protozoa. But, hey, we have a big audience.
We do.
Yeah.
And a knowledgeable audience at that.
And there's a big universe of things left to be discovered, so...
Hear, hear.
Right.
Yeah, we should say that we don't know of any known of these things, but maybe they'll be found, yeah.
Right.
Right.
And then viruses that infect mitochondria or chloroplasts.
Yeah, that's a great question.
These are all great questions.
I don't know of any that do.
And also evidence for endogenous retroviruses in the mitochondrial or chloroplast genomes.
Now, I think we can. I think I can pretty much safely say that there's no ERVs in the mitochondrial DNA that I know of. I don't know about chloroplast genomes. But mitochondrial DNA, at least, I don't know that I've specifically. I certainly have specifically queried it for ERVs, but I think mitochondrial DNA is pretty much accounted for.
I think they have phage-like sequences in mitochondrial DNA.
Okay.
Because they used to be bacteria. So you can find traces of that. I mean, they're not very big, the mitochondrial genomes, right?
Right.
But I do think there are
Chloroplasts are
some...
bigger.
yeah?
Much bigger.
I do think that there are phage sequences in there. What do you think about this alltrials.net?
Cool.
I think it's a good idea.
I don't think that everybody's going to subscribe to it. Do you?
No, but I think it's pretty huge that Glaxo has done it.
Yeah.
Yeah, I think it's sort of an interesting follow-up to the previous letter. I think it's interesting that I don't know anything more than what's just written here, but I think it's nice that Goldacre, in addition to apparently exposing a lot of what he feels is bad stuff, will actually do something positive. Like
he is proposing a solution, maybe only a partial one, but certainly,
I mean,
I think generally, if you're going to complain, you really ought to come at it with a solution in hand, and I think that's nice, and I think it's great that GSK has signed on. So we'll see.
So this is related to our earlier discussion of Roche and the Tamiflu clinical trial results, right? Remember?
Yep.
Right.
So I think Roche is not going to sign up for this all trials.
Well, it depends. I mean, what's being asked by the Cochrane collaboration, as I understand it, is they want the data. All trials may simply, I mean, if there's an option of simply publishing a report as you would a publication.
Right.
That's a horse of a different color.
Yeah.
And if you have a place where you can publish negative results, this is a huge and long-standing problem that we've talked about before. Nobody publishes negative results. So if your study was just insufficiently powered to reach a conclusion, then it goes in the file drawer.
It seems to me that this is all a first step towards getting all this stuff out in the sunshine.
Yes.
So this website is basically a petition that you sign, and you can donate money if you'd like. That's all it is at this point. So it's not any kind of initiative other than saying it's time that all clinical trial results are reported. Sign here if you support this. And so GSK sign, that's cool. But I don't know if in the end anything will happen of this, right?
The follow-through, I think the appropriate follow-through would be some kind of a publication mechanism.
Yeah.
So that you could have peer review of negative results.
Right.
And that's really what's been missing.
Well, then imagine all the data miners out there.
On the page itself, on the right, there's some Twitter information, and there's something that was posted 3 hours ago. Roche makes new transparency promises, re-Tamiflu, and then the next thing is researchers not impressed. So those of you that like to follow those kinds of things down the hole, you can do that.
You like sitting next to me, Dickson?
I do, actually.
I invited Dickson to sit next to me so he could see the show notes for once in his life.
And he's not offended by my being here.
I'm not offended, but you can see. Isn't it cool what we do here with these notes?
Of course
you should get a computer and you could do this.
I have one. I brought it in. I just haven't hooked it up yet. Come on. I've only been here for how long?
Okay.
Well, he had to bring it in with that hand truck because it's the size of a refrigerator.
It's an iPad.
It's got vacuum tubes.
Yes.
Stop it.
All right, the last email is from Christina, who writes, Dear TWiV, PM and more. Dear TWiV, PM and more. Right. Maybe more. Who knows? Thank you for making our lives better. I've been finding a refuge of sanity and happiness listening to you for a long time. I have a plea for help. I'm a graduate student in neuroscience. Thanks for all the viruses. And when I walk, like many others, look for some science. I thought I would learn something about cellular biology to understand you guys better. I bought The Wisdom of Your Cells by Bruce Lipton. Glowing reviews on Amazon, Audible, everywhere, but the book itself is piled with new age phrenological quackery. The book compares the author to Rachel Carson and Stephen Hawking, according to now very ironically named publishing company Sounds True. How can we fight this kind of science noise? In retrospect, I should have known better. The word wisdom should have tipped me off. If we peer review the science we publish for each other or put it on archive, why is it okay to broadcast nonsense to the rest of the world with the, quote, endorsement of science, unquote? Do you guys know anything we can do as scientists to stop this kind of thing? News stations cannot tell open lies. There is some expectation that what is printed for the public is in some sense true. But when it comes to popular science, there seems to be no limit. Do you do anti-picks?
We just did one.
I have spent a lot of money buying popular science audiobooks. and I have never found anything nearly as good as your podcast in terms of being true. It felt like I was briefly in the dark ages.
Oh, wow.
I should just listen only to Twix.
Here, here. And between.
I think anti-picks is a great idea.
Yeah.
All right.
The problem is that they would backfire.
How so? By giving them publicity? Yeah.
Yeah.
True.
Probably.
And for many of the things that I would pick as anti-picks, there is no such thing as bad publicity.
Yeah. That's right. That's
Just don't spell your
right.
name wrong, right?
Yeah.
There's really nothing you can do because we
Nope.
have freedom of publication, right?
Right.
And we can't cover everything here on Twix, but, we can criticize things. And you just have. So people, if you see things that you want to warn people from, you can tell us. We have some reach, right?
Yeah, and when, you can talk to your friends.
Yeah.
Right.
And
Local
post some
info.
non-glowing reviews on Amazon.
Right.
Well, I
Or
can
to
show you a
just
few of
avoid
those.
buying them in the first place, you could check out the authors, on Wikipedia or Google them and see
Yeah.
what you can find out about their background.
Right.
Yeah, that's one of the first things that I do when I look at any of these kinds of things is check on the background of who's
did you
the
put
author.
this wiki link in?
Yes, I did.
Oh, Bruce Lipton is an American developmental biologist who is best known for promoting the idea that genes and DNA can be manipulated by a person's belief. I
He
wouldn't buy
teaches
it.
at the New Zealand College of Chiropractic.
I wouldn't buy this guy's books.
I just there's a whole series of red flags in that sentence,
Oh my
Correct.
gosh.
Correct.
or in those
American.
two sentences.
Oh, well.
Yeah.
So, Christina, people can publish whatever they want if they find a publisher, but
Or they can self-publish.
They can self-publish, but you can always come to Twix.
Remember,
Hear the
science
truth.
is a self-correcting process.
It's
So it doesn't matter what you publish. Eventually, it'll be either proven or disproven.
Yeah, but we have to fight people who write like this.
Yeah,
and
I know,
Write
I know.
quackery. Phrenological quackery. What a great name. Not only that, New Age phrenological quackery. I wish we could use that as a title, but
People would get the wrong idea.
Get the
Yeah,
Aflac
exactly.
Doug to sponsor it, maybe.
all right. Let us do some picks of the week. Dickson, you probably don't have a pick,
I
right?
picked these two articles.
Can't pick articles. There's so many things you cannot do.
No, I'm gonna pass on the picks this
You
week.
did last week, too. You know what that means. Two
Did I?
passes and you're out.
Really?
No, Dickson. I wouldn't ever do that to you.
Thank you.
Kathy, what do you have?
Well, I have something that my other brother sent me, and it's a link to a blog post, and within that there are several links to a video and so forth that you might want to look at. And the sort of the long version recap, or the technical one, is that graphene, a very simple carbon polymer, can be used as the basic component of a supercapacitor, and it goes on for lines and lines. And then it has English translation. He painted a DVD with a liquid carbon solution and stuck it into a standard issue DVD burner. The result, absurdly cheap graphene sheets, 1 atom thick, which held a surprising amount of charge without further modification. And so that was evidently reported a year ago, and there was a viral video making the rounds quite recently. But then recently they have a paper coming out in Nature Communications describing a way to make it mass-produced, so possibility of mass production. And all of this is important because this could be new and better batteries.
Nice.
Got it.
So
But they're not
Very
made
cool.
of. They're
Cool.
not made of viruses, though. They're made
No.
of graphene.
no, they're not made of viruses. They're made of graphene, single layer carbon. So
Well,
pretty cool.
it's as the article points, as this article points out, it's a very early finding, but it would be great if this works.
yeah.
Alan, what do you have?
I have a YouTube video that I found just a little while ago. So if you click it, it'll autoplay, annoyingly, I'm sure. But this is from the Michael J. Fox Foundation, and it's just a brief description about how drug development works. And it's a very lucid, well-illustrated, well-filmed explanation of how you get from basic research about a disease or about anything to a point where you're putting pills in a bottle. And I just thought that it was very well put together and did an excellent job of summarizing what is an extremely complex process.
Nice.
It is cool.
Cool.
I
Excellent.
like the writing speed it up. It's really cool.
Yeah, it's become a popular animation technique, the sped up drawings, and the artist does a nice little cartooning job and illustrating the Valley of Death in clinical trials and all this.
It's great. Very nice.
Gets across the main
Excellent.
concepts.
Beautiful. Rich, what do you have?
Well, I've been saving this for a time when I really didn't have a pick.
This is great, though.
And I ran over
I
this.
share this
ran
with you?
across this, I don't know, a long time ago. It's a video that's the explosive breach of the Condit Dam. Okay, it's on Vimeo. And the Condit Dam, I think, is on the Salmon River in Washington, and
White
part of an ecological project, I suppose. They decided they didn't need the dam anymore. They wanted the river back.
That's right.
And so they blew up the dam, and they videoed the whole thing. Actually, they blew up a part of the dam. It's not like the whole thing collapses. But I'll tell you, it's pretty impressive because it floods the downstream part and empties the upstream part in a big hurry. And they've got it all on time-lapse photography. It's pretty cool.
Yeah.
It's amazing.
Wow, look at that.
Yeah.
Oh, look.
Yeah.
There goes the only dam that you could say Condit wasn't responsible for.
Look at that black stuff. Wow.
Yeah.
Yeah.
Outstanding.
Amazing.
The power of nature.
Just a little hole in the bottom.
Well, the power of dynamite.
It was just sitting there
If you
waiting
listen
for
carefully
this.
as it's in the beginning of it, before it starts, you hear somebody shout, "Fire in a hole!"
Right.
And then, on the downstream side of the dam, you can see these little people coming out to these viewpoints and up above the dam. I'm not sure I would have done that. And then they run back out. Yeah.
Tsunami, right?
It's frightening.
Yeah.
It is frightening in some ways.
So you've known about the Condit Dam for some time, Rich?
No. I just I don't know who ran across. I don't know how I found this. This is the only thing I know about the Condit Dam.
Look at the water up here going down.
Oh, yeah.
Wow.
True.
And now they're going to have new life down here, right, Dickson?
They will.
Little ecology.
No. What they're going to do is allow the salmon to migrate up that river further.
Neat. Oh, it's all eroding away. This is spectacular. Very nice, Rich. Lovely.
Yeah, so over a period of time, the river will return to its original state.
Neat.
Yeah.
Correct.
Actually, this was a while ago. It'd be interesting to have some sort of and time lapse of what it looks like now.
Sure.
Well, my pick is a little more serious. It's hard to follow that.
The tree of life.
It's a blog post by Jonathan Eisen, who is a microbiology professor at UC Davis, big advocate of open access. In fact, his brother is one of the co-founders of the PLOS journals.
I just interviewed him recently.
Oh, yeah? Jonathan or his brother?
Jonathan.
Yeah. And he, this is called 10 Things You Can Do to Really Support Open Access. And this is about, 10 things you can do, all right? Like only publish in open access journals. Do not do any work for non-open access journals. That includes reviewing, suggesting reviewers, etc. Cancel all subscriptions. Embrace openness in your academic work. And one of the things he says is reward people in job hiring, merits, and promotions for their level of openness. Do not reward them for closed activity. So it means if you have 10 cell papers, you don't get tenured.
Right.
Lobby for more open access. So, I mean, these are really serious things that are not going to happen anytime soon.
Right.
And James Chen, for example, just published 2 back-to-back papers in Science, and you could not convince him to publish them elsewhere, I'm sure, because he wants to renew his Howard Hughes appointment when it comes up for renewal, and he needs 10 Cell and Science papers, So I think these are great, but tough.
Well,
right?
But the reason it's not going to happen is because people aren't doing it. And that's kind of self-fulfilling. If I think Jonathan's argument here is if people really were serious about this and enough folks, a critical mass of folks really said, okay, I'm going to publish only in open access journals. I mean, if Chen said that I'm going to send these back-to-back articles to PLOS,
right,
then that's where they'd be, and they would still be really great papers, and we'd still be talking about them on TWiV, but our listeners would actually be able to see them.
Yeah.
And that would be a couple of I don't think Chen's going to have a problem getting his Hughes position renewed based on his work.
No, but he publishes in
He publishes in
journals, yeah.
top journals.
I mean, what if we only did open access papers on TWiV, for example? We'd miss a lot of good stuff, right?
Are you implying?
Yes.
Are you implying that the open access journals publish articles of less significance?
No, it's just we would miss the
we've
broad.
We've talked about a lot of open access papers on here, but there's good stuff still coming out in closed journals.
I think we each have to make an effort, but there will always be people who want to sell Science or Nature paper, no matter what.
Yep.
Until it gets to a point where you there's going to be some point where that is no longer the route to fame. And
let me take a little informal poll here, okay? Rich Condit, are you going to publish in only open access journals for the rest of your career?
No. Well, it's we're talking about a pretty short horizon.
Doesn't matter.
No, that's not true. As a matter of fact, I work for a journal that is not an open access journal.
Okay. How about you, Kathy?
Ditto. The Journal of Virology is in the ASM journals. You know, ASM has the MBio as their open access journal, but some things are going to go to JV.
And are you going to ask me?
Sure.
Yeah.
Hey, Vince, what are you going to do?
The other day I had this discussion with my postdoc who, wants a Cell or Science or Nature paper, and then she said at the end, well, maybe we'll send it to PLOS instead. That's good, right? I said, yeah. Mm-hmm. So I think you got to work on it little by little, and but there's still this culture, right?
Yeah. Well, and I'll go ahead and comment too, even though I'm not doing research in the lab, I write for
that's right. Closed access.
In fact, the interview that I did with Eisen was for an article that I was working on for Science. Now, it happens to be a section of Science that is that's freely accessible, but still, I'm working for the man.
Alan, let me ask you a question.
Sure.
So, Science magazine has all this this commentary and news in the front, and then the articles, right?
Yeah.
Well, let's say if they made all the articles open access, but still sold subscriptions to the paper journal, and people could get all this extra stuff online by having a subscription, would that work? Or would people drop their subscriptions because the articles are open access?
I think people will drop their subscriptions because the articles are open access, because the reason for most subscriptions to Science is not the stuff that I'm writing. It's the science. So that's where all the institutional money is. That's where most of the individual subscriptions are for the research articles.
The perception is that the higher the prestige of the journal, the more difficult it is to get your paper published, and therefore it's more recognition for the work that you've done. How do you convince a tenure committee or a granting committee that that's not the case? And everybody lives in fear of that. I mean, I did all my life. You try for the highest journal possible with the most difficult editorial board, and if you get in, everybody says, ooh, wow, you got into...
Of course.
And if it's an open access journal that publishes everything...
Well, but
it's peer-reviewed. It is peer-reviewed, I agree.
Let's get something straight here. Open access does not mean PLOS One.
Yeah, but everybody thinks that, though.
I know, and that is a big part of the problem. A lot of open access, the main PLOS journals, apart from PLOS One, are traditional journals.
Right.
The only thing that is different about them is that the authors are paying for the publication rather than the subscribers. Mm-hmm. So that model doesn't say anything about the toughness of the peer review, and people get papers rejected from the PLOS journals all the time.
Sure.
The PLOS One has a different approach, quite deliberately, that they have decided that they're not going to review for this sort of significance standard that Nature et al. Prefer. That the peer review is only to ensure that the data match the conclusions, basically.
Well, Alan, several of the papers that I submitted to Science in the past, I've had several publications in Science, but I had many more that were reviewed favorably and said this is too narrow a subject to be published. And the Science is great, but so there's no room for a larger amount of Science if it's a paper publication. If it's an online publication, you can put as many as you want in there. So maybe the argument should be that they should all be online only, and then the restriction of pages is moot.
Well, right. That's that would help, but I think that's a separate issue. The relative prestige problem is serious, but I think it's just a perception problem because there are open access journals that are hard to get into.
You didn't interview Eisen about open access, did you?
No, I talked to him about microbiomes.
Yeah. I'm surprised he let you interview him for science.
There is a little bit of irony here.
Hey, well, he has this blog post that says don't do anything for, closed access journals, and then he gave you, not that there's anything.
But refusing to talk to a reporter would be pretty closed of him, wouldn't it?
Yeah, but he knew you were doing it for science, right? I assume you told him.
Sure, and in fact, after we did the interview, I sent him a thank you email, and he sent a note back saying, by the way, if you want to use the recordings or anything else and publish those, that's fine too. So he wants everything to be totally open, and talking to reporters is an open thing. So I think it's a different issue.
Yeah. So just one more question about all this extra value material in science and that sort of thing. Well, why do they do it if it couldn't sustain the journal on its own? Is it just extra added value, and does that drive people to subscribe? I wouldn't think it would drive an institutional subscription, right?
It doesn't drive an institutional subscription. It does attract readers. It does attract views on the web. It is something that readers like, and science, remember, is part of the AAAS.
Right.
So as an organization, they want to promote public understanding of science. Personally, I think it would be better if they made their news articles free, and that would promote better public understanding of science, because those are often the perspective articles too.
Yeah.
Those are often the more approachable parts.
And the editorials.
But the issue for organizations like Nature, like Science, and Nature is a purely commercial venture, part of Macmillan. Science is part of a nonprofit organization. They're the publication for them. But in both cases, there's this huge institutional infrastructure built around subscriptions.
Yeah, sure.
And it's all well and good for Jonathan Eisen or you or anybody else or me to say, well, all the science should be open. But then you've got to figure out how you get from A to B.
Yeah. You've got another...
That's the hard part. I think there's an understanding at... I know there's an understanding in the Nature offices. I believe there's an understanding at Science, and it may even be trickling to Elsevier eventually, but I wouldn't count on it, that open access could be a viable business model.
Right.
But they just can't quite see how to get there from where they are right now, because, what do? Do you prorate all your subscriptions, refund the money, and how are you going to cover the gaps in what are you going to do to get from here to there? I mean, it's really nuts and bolts stuff.
So I just wanted to comment, because you were commenting on why people subscribe to the hard copy version of Science. And even though I have completely free access to it through the university library, I still get the hard copy. And it's in part because I like to read the stuff up in front, and I like to scribble all over the articles that we just read, for example. But one thing that they do here to promote open access publishing is help defray the costs of the page charges for open access journals for investigators, as a way to promote that other business model of the open access journals. So I don't know if that would be. It doesn't. I mean, there's not an easy way to solve the problem that you were describing, Alan, in terms of the business model of subscriptions and paying for the journals.
Right, and I think there's a feeling. Well, I know there's a feeling in the industry that the open access model is working right now, but or is more or less working right now, but it's still new enough that it's not obvious that that's a sustainable way to run a business.
Yeah.
There's one more point to make, too, and that is that the public has a right to know the results of research that they've funded.
Sure.
And all that money comes from them. So how do they have access to this knowledge otherwise? And they should have access to it.
Well, now, the administration just announced that everything funded by the U.S. has to be published within a year, open.
Really?
Right.
Open within a year, though. I mean, so that's the answer for now, Dickson. A year later, you can see the work that you funded. but I think it should be.
I mean,
soon.
last year's journals can be open. And
Yes,
this year's
yes.
Right.
Yes.
Yeah, and ASM does it after 6 months, I think, right, Kathy?
Yeah, I think
That sounds right. Yeah.
But with ASM, you do have to pay page charges anyway, and they're pretty high, just like the PLOS journals. So
Yeah.
And classically, the answer to this question, how do the taxpayers get access to the research, was you go to the library.
Right.
And you can.
Well, that's right.
It'll take you 6 months through interlibrary loan to get all these at your local library, but you can get these papers. The only reason we're having this discussion about open access now is really that we're at a point where we can get beyond paper.
Well, that's a good reason, though.
It's a fine reason.
Yeah, okay.
But it's just important to keep a perspective that this is this is something that is not locked away from the public. It's just not accessible online.
Right, but it's a good thing to have, I think. I mean,
Yes.
we can get papers right away, and I think the public should be able to do that. And someday someone will figure out how to do it.
And I agree, and I think this is the future of the science publishing industry. And they'll still be able to pay people like me, by the way, because paying with page charges, I don't care if it's coming from page charges or subscribers.
Right.
My checks are clearing. I'm happy.
Yes.
Okay, I'm not quite that cynical, but so that's not the issue. The issue is how do we get to that point? And there are, as you pointed out, institutional barriers with people wanting a Cell paper and a Nature paper, and there are these corporate barriers with the companies that publish the journals needing to figure out how to continue publishing the journals.
Right. See, that's what a pick of the week can do, Dickson.
It spawned an entire
It's all
conversation.
right. I don't think people mind that. It's
No,
an
no, no,
interesting
I think-
issue. We have 2 listener picks, and they both relate to computer viruses. And first is from Mark via Twitter. This IEEE piece on the Stuxnet computer virus might interest TWiV listeners. Entry, pathogenesis, replication.
Yes.
Wow.
The real story of Stuxnet. This is the malware that stymied Iran's nuclear fuel enrichment program.
At least temporarily.
Yes.
Yes. So, it's a nice article on how
Yeah.
that worked, and with analogies to biological viruses. Thank you, Mark. And then from John, dear Twivers, I thought this would make a fun reader pick of the week. It's the story of a completely mechanical kind of virus-like spread of a property, really more like a prion than a virus.
This is great.
This is 1997 outbreak of virus infecting 0.050 pin D connectors.
Oh my God! Not that again.
Do you know what a D connector is?
Of course
Dixon?
I do.
This right here. That's
It's
a D.
one that proceeds C connectors.
Follow C connectors.
No, it's shaped like a D when you look at it sideways.
Is that what
See that? You turn it sideways and it looks like a D.
It's like these old printer cables you choose to have. Well, Dickson's computer probably still has. That's
I
got
have a
it's got
hamster
whole-
that runs on a generator.
Right. It's got a whole bunch of pins inside it, and you stick them into a whole bunch of holes on the other side. And this is a disease that it affected these connectors.
Yeah.
A sexually transmitted disease.
Yes, it's very funny. So you should read that. So that's great. It's an oldish article, but it's still cool. So isn't that funny? We had 2 virus picks of the week, and they're not real viruses. They're
That's
computer
right.
viruses.
That's right.
I did an interview the other day on WNYC, and the guy said, what's his name? Kurt Anderson?
Yes.
He said to me, don't you feel good that the computer world took virus from you? And that was me. Like, is it from me? I don't know about feeling
Now-
good, but yeah, we were there first.
I have to I have to let the rest of our panel know that I'm now privy to an observation that only Vincent was privy to before, and that is the wear marks on his typeboard. Your keyboard wear marks suggest usage frequency, and that's very unusual because the top row. What are the most frequently used keys on your computer? And you would never guess what they are unless you had a way of wearing off a surface. And Vince's keyboard has wear marks as to the most frequently used keys on his computer. That's fantastic.
Yeah, it's
Vince.
VINCENT.
Yeah, that's well for virus.
I was thinking the letter F probably got quite a-
Well, there's a lot of that here too. You want me to read them off to you in a way that doesn't spell something?
The shift key is worn in the enter, and then ASD and E and C And then
That's
L.
quite-
Then L at the other end.
I
don't
got
know
about
why.
the same pattern here.
You
Yeah?
have a very hard. I can hear you typing all the way out in the hall.
It's because you drive me crazy.
Vincent does. He does pound the keys.
I do pound the keys?
You pound
Yeah, you
them. You
pound the
pound
keys.
them.
Well, I'm going to take a picture of this and I'll put it in the show notes.
That's fantastic. And I think it's great.
You want to sit next to me every week?
I do.
Oh, thank you. You can find TWiV at iTunes, twiv.tv. And if you do like us, what you can do to help is to go over to iTunes, subscribe, and leave some kind of rating. When you rate podcasts, it helps to keep them visible in the directory over there. And, of course, send us your questions and comments to TWiV at twiv.tv. Dickson Despommier, thank you for joining us.
My pleasure.
Are you sure?
Absolutely.
You seem reticent.
No, I'm thrilled.
Next week you'll be in Germany, right?
That's true.
All right. Maybe we'll see you afterwards.
No, not Germany next week. Next week is England.
Okay. Verticalfarm.com, trichinella.org,
Yes.
and medicalecology.com.
Yes.
Kathy Spindler is at the University of Michigan. Thank you, Kathy.
Thanks. This was fun. And as always, I learned a lot.
And Rich Condit is at the University of Florida, Gainesville. Thank you, Rich.
Where it is 59 degrees and cloudy.
Go
We
to-
didn't do the weather today.
Yeah, a little bit. We did. Kathy talked about the snow.
Yeah.
Did that get taped?
It did.
Okay.
It actually got digitally recorded.
Oh,
We don't
okay.
use tape anymore, Rich.
I'm sorry.
Rich, are you going sailing this weekend?
No, I'm leaving tomorrow for Oregon. I'm going to the snow, and I will be gone for 2 weeks.
You're skiing?
Yes, as a matter of fact, I'm going to see if this old guy can learn to snowboard.
Good luck. Don't get hurt.
Wow.
I'll try not. We'll see what happens.
Have fun.
Thanks.
Alan Dove is at alandove.com and on Twitter. Thank you, Alan.
Always a pleasure.
You'll be here next week, right?
Yes, I plan to.
Be you and Kathy. Is that right, Kathy? You're here?
Yes.
All right, the three of us.
Cool.
That'll be cozy. Right, Dickson?
Yes.
I'm Vincent Racaniello. You can find me at virology.ws. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.